आजको हाम्रो स्पिकर हुनुहुन्छ डाक्टर बिलोन खम्बु, उहाँलाई स्वागत गर्न चाहन्छु। आज हामी एउटा टपिक जो अलिकति हाम्रो लागि अलि नौलो, अलि एक्जोटिक नै छ, नयाँ टपिक हो। हुन त 2016 मा यो अटोफेजी भन्ने जुन शब्द छ, यो शब्द बायोलोजिकल साइन्सेसमा नौलो त होइन, तर पनि जनमानसमा अलि नौलो नै छ। यसलाई मैले हिजो फेसबुक पोस्टमा लेख्दाखेरि अटोफेजी भनेको के हो भनेर भन्दाखेरि यो चाहिँ हाम्रो घरको भ्याकुम क्लिनर जस्तो हो। घरलाई बेला बेलामा सफा गर्नुपर्ने हुन्छ। यदि हामीले बेला बेलामा सफा गरेनौँ भने त्यसको फोहोरको थुप्रो बढ्दै गएर घर नै दूषित बनाउने र गनाउने र बस्न अनुपयुक्त हुने सम्भावना हुन्छ। त्यस्तै काम हाम्रो शरीरको सेल्सहरूमा भइरहेको हुन्छ, अर्गनहरूमा भइरहेको हुन्छ र त्यसको बारेमा अहिले गहन अध्ययन अनुसन्धानहरू जारी छन् र त्यही अध्ययन अनुसन्धान गर्ने एउटा व्यक्तित्व हाम्रै समुदायको, हाम्रै डाक्टर बिलोन खम्बु आज हामीहरूको बिचमा हुनुहुन्छ। उहाँले हामीलाई रोल अफ अटोफेजी इन लिभर डिजिजेस को बारेमा कुराहरू गर्नुहुनेछ। बिलोनजी काठमाडौँबाट, भक्तपुरबाट हुनुहुन्छ। उहाँ चाहिँ भक्तपुरको मध्यपुर, अहिले मध्यपुर नगरपालिका भएको छ क्यारे, मध्यपुरमा जन्मनुभयो, त्यहीँ हुर्कनुभयो, त्यहीँ स्कुलिङ गर्नुभयो। अहिले अमेरिकाको न्यू अर्लिन्समा टुलेन युनिभर्सिटीमा एसिस्टेन्ट प्रोफेसरको रूपमा काम गर्दै हुनुहुन्छ। उहाँ चाहिँ आफ्नो परिवारसँग, आफ्नो श्रीमती, 8 वर्षको बाबु, 8 महिनाको छोरीसँग त्यहाँ न्यू अर्लिन्समा बस्नुहुन्छ। यो चाहिँ लुइजियाना राज्यमा पर्छ। न्यू अर्लिन्समा मुभ गर्नुभन्दा पहिला बिलोनजी इन्डियाना युनिभर्सिटी स्कुल अफ मेडिसिन, जुन चाहिँ इन्डियानामा पर्छ, त्यहाँ पोस्टडक हुनुहुन्थ्यो। पोस्टडक्टरल ट्रेनिङ त्यहाँ गर्नुभन्दा अघि पिएचडी को डिग्री बिलोनजीले क्योटो युनिभर्सिटी स्कुल अफ मेडिसिनबाट लिनुभएको छ। जापान जानुभन्दा पहिला बीपी कोइराला इन्स्टिच्युट अफ हेल्थ साइन्सेस स्कुल अफ मेडिसिनबाट मास्टर्स गर्नुभएको छ। त्योभन्दा पहिला माइक्रोबायोलोजीमा बिएससी गर्नुभयो त्रिभुवन युनिभर्सिटीबाट। र यो यात्रा भक्तपुर, मध्यपुर हुँदै त्यसपछि जापान, त्यसपछि इन्डियाना, त्यसपछि न्यू अर्लिन्सबाट उहाँ अहिले अटोफेजीमा रिसर्च गर्ने एउटा इन्डिपेन्डेन्ट रिसर्चर, टेन्युर ट्र्याक फ्याकल्टीको रूपमा काम गर्दै हुनुहुन्छ। उहाँलाई हामी आज पाउँदाखेरि हामी एकदमै गौरवान्वित छौँ र हाम्रो अनुरोधलाई जुन स्वीकार गरिदिनु भयो हजुरले, त्यसको लागि हामी धेरै ग्रेटफुल छौँ, कृतज्ञ छौँ। र बिलोनजीको रिसर्च चाहिँ 3वटा मेजर टपिकमा फोकस छ। यसमा उहाँले अटोफेजी एन्ड लिभर मेटाबोलिजम त्यो टपिकमा काम गर्नुभएको छ। एउटा सब टपिक त्यो छ, यसले कसरी लिभर मेटाबोलिजममा इम्प्याक्ट गर्छ भन्ने। अनि सेकेन्ड टपिक छ रोल अफ अटोफेजी इन डिजिज, डिजेनरेसन एन्ड रिजेनरेसन। यो विशेष गरेर लिभरमा बढी एप्लाई गर्छ, त्यो बढी रेलेभेन्ट छ। लिभर चाहिँ एकदमै हाइली रिजेनरेटिभ अर्गन हो, त्यो पनि हामी अलिकति सुन्नेछौँ। त्यसपछि मोलिकुलर मेकानिजम्स अफ अटोफेजी ड्युरिङ सेलुलर स्ट्रेस, यो चाहिँ हुन त उहाँको कन्टेक्स्ट चाहिँ लिभर छ, तर यो चाहिँ अलिकति ब्रड हो। यसले कसरी यसका चाहिँ मोलिकुलर प्रोसेसहरू के हुन्, यसमा कुन जिनहरू इन्भल्भ हुन्छन्, कुन प्रोटिनहरू इन्भल्भ हुन्छन्, यसको चाहिँ भित्र इन्जिनभित्र भनौँ, अब इन्जिन भनेर मैले एउटा एनालोजी दिन खोजेँ, तर यसको डिटेल प्रोसेस के हो त्यसको बारेमा हामी आज सुन्नेछौँ। अब मैले यति डाक्टर बिलोन खम्बुको यति इन्ट्रोडक्सन गरेँ। अब विथआउट एनी फर्दर ड्यू म डाक्टर बिलोन खम्बुलाई स्वागत गर्न चाहन्छु। बिलोनजी, माइक इज विथ यू, वेलकम।
Thank you for such a wonderful introduction. दाजु यो स्क्रिन शेयर गर्दा हुन्छ?
हजुर, you are allowed to share your screen. Please go ahead.
Hello and namaste everyone. साथीहरु म यो रिसर्चको विषय भएकोले म आफ्नो सबै रिसर्चको स्लाइडहरु अंग्रेजीमा नै प्रस्तुत गर्छु। मैले तारा दाइसँग कुरा गरिसकेको छु। तपाईहरुको केही क्वेशनहरु छन् अथवा के मैले नेपालीमा क्लेरिफाई गर्नुपर्ने छ भने हजुरहरुले नहिचकिचाइकन मलाई बिचमा रोकेर क्वेशन सोध्दा हुन्छ। कति कुरा अब यो रिसर्चको साइन्टिफिक टर्महरु नेपालीको ठ्याक्कै रूपान्तरण नभएकोले म जेनरली यो स्लाइडहरु चाहिँ अंग्रेजीमा नै प्रेजेन्ट गर्छु, तर क्वेशन एन्ड आन्सर चाहिँ अंग्रेजी अथवा नेपालीबाट हामी गर्दा हुन्छ भन्ने चाहिँ सोचेछु। आजको मेरो बेसिकली friends, welcome to this seminar. The title of my today's talk is Role of Autophagy in Liver Diseases. So before I really start my talk, I would like to thank NIRI for giving me this wonderful opportunity to showcase my lab's research work. In today's talk, basically, I'm going to tell a short story about 3 different things. One is about how liver disease develop in a pathological perspective. And the second is how we study the liver disease in a preclinical model in a basic research lab. And the third thing also, how the autophagy deficiency plays role in the development of liver diseases. So those are the 3 things I'll try to cover under today's talk. So these are my disclaimers. I have no conflict of interest nor commercial interest to disclose. And I'm not a clinical doctor, so any questions related to liver disease need to be consulted to the physicians. And these are the outline of my today's talk. So basically, I'm gonna start with the basics about the liver structure and the functions. Then I will show what are the pathological changes that happens in the liver diseases. And with that, I will show the scientific premise on which we base our ongoing research work. And with that, also the different kind of preclinical models that are used to study the liver diseases. And under that preclinical model, also a genetic model that we frequently use in our lab to study, that's called autophagy deficient liver. So under that model, I'll give a brief background about the autophagy process. And basically what I'll try to tell you under this section is what happens when autophagy is defective in the liver. So many things happens in the liver when autophagy is defective, but today I'm gonna specifically focus on injury associated regeneration that happens in the context of autophagy deficiency. So let's get started with the basics about the liver structure and the function. So as we all know, liver is one of the largest and the biggest organ of human body that's located in the upper right side of the abdomen below the diaphragm. So it has different functions ranging from carbohydrate, lipid, or protein metabolism. The liver also detoxifies the drugs or the xenobiotics. Liver can synthesize different plasma proteins and secrete it into the general circulation. And one of the very popular functions of liver is to synthesize the bile acid. As we all know, bile is synthesized by the liver, then it's secreted out from the liver and temporarily stored into the gallbladder. And with the intake of food, then the bile acids are squeezed and secreted from the gallbladder out into the intestine. And in the intestine, this bile acid is necessary for the absorption and the digestion of the lipids. So at a very cellular level, the liver consists of different types of cells. However, one of the very important cells in the liver is the hepatocytes. That makes up the 80% of the total liver volume. And other cells that are present in the liver are endothelial cells, stellate cells, or the immune cells, such as the Kupffer cells. So when we see histologically, one of the interesting things about the liver is that liver receives a dual circulation. That means liver receives oxygenated blood from the hepatic artery, and liver also receives a low oxygenated but a nutrient-rich blood from the intestine through this portal vein. So here I generally try to show you the histological section of the portal triad. What does that portal triad means is that there are 3 different vessels. One is for the portal vein, through which the liver receives the nutrient-rich blood from the intestine. Another is the branch of the hepatic artery, through which the liver receives the oxygenated blood, and another one is the bile duct. So through this bile duct, actually the liver secretes this bile acid into the gallbladder. So you can clearly appreciate that this region is called portal triad. And from the portal triad, there is a lining of the sinusoids along which there lies the many hepatocytes in a very linear fashion, which we can see here as a form of a sheet of hepatocytes running from the portal triad up to the central base. So in this general liver section, you can clearly appreciate that most of the cells are the hepatocytes, and there are very few of the non-hepatocytes like the endothelial cells, stellate cells, or the Kupffer cells. So this is the general histology of a normal liver. Now, let's see what are the pathological changes that happens in the liver during the course of liver disease development. So the fundamental idea is very simple in the liver. What happens is a normal liver, when exposed to different hepatic stressors, the hepatic stressors could be high fat diet, alcohol, or the genobiotics. So the first thing that happens in the liver is because of this stressor, there is a development of steatosis. What does steatosis means? It's actually accumulation of fat in the hepatocytes. Here we can see there are a lot of white bubble kind of structures. Those are the fats that are being deposited within the hepatocytes. So with the development of steatosis, then the next thing that happens in the liver is called a steatohepatitis. That means there is not only accumulation of the fats, but there is now infiltration of different immune cells. That we call it as an inflammation. So that's why the combination of steatosis and the inflammation, it's called a steatohepatitis. So after this development of steatohepatitis, then there is a next phase of pathological change, which is called a cirrhosis or called a fibrosis. So basically the fibrosis and cirrhosis means the liver stiffness increases because of accumulation of extracellular collagen fiber between the hepatocytes. So one of the very interesting thing about these pathological changes, which I would like to take your attention, is that no matter what kind of hepatic stressors are there, maybe could be fat, could be alcohol or genobiotic, they all cause the same kind of pathological changes. Always start with the steatosis, then steatohepatitis and the cirrhosis. So that's why our research interest in my lab is actually to understand the molecular drivers of these liver pathologies, and we wanted to see whether they could be therapeutically targeted or not. Now, based on that, our scientific premise about ongoing work is that there is a normal hepatocyte in the liver. When it's exposed to the different stressors like high-fat diet or alcohol or genobiotic, what happens is the hepatocytes are stressed, basically, and they release different hepatic factors. We call it hepatic factors because these are the factors specifically released from the hepatocyte. So, and the factor could be either X or Y. That means it could be nuclear or it could be a cytosolic. Sometimes the hepatokine word is also coined. So idea is a stressed hepatocytes release these factors outside the hepatocytes extracellularly. So when the stressor level is crossing the threshold limit, then in that context, the hepatocytes can also undergo cell death process, or we call it as a cell injury. And in that case as well, the hepatocytes release different intracellular components that we call it as a damage-associated molecular pattern. It's called a DAM. So no matter whether the hepatocyte is stressed or the hepatocytes are dying, they release the hepatic factors. And these hepatic factors are actually responsible for activating the immune cells, such as macrophages, to cause the inflammation. These hepatic factors can activate the progenitor cells to stimulate the liver regeneration, or these factors can activate the stellate cells, which are a particular type of cell in the liver that are responsible for the deposition of collagen fiber for the formation of fibrosis or the cirrhosis. And we believe that the repeated this kind of insult results in the development of the tumor. And that's why our long-term goal in the lab is to understand these hepatic factors. Basically, we want to identify and therapeutically target these hepatic factors because we believe that these are the hepatic factors that are responsible for the development of all of these liver pathologies. So now let's see how we study the liver pathogenesis in a preclinical model. So when I say it's a preclinical model, that means it's before the clinic. It's in a basic research lab. So we always start with the in vivo mice model. So we have a C57 black mice. So we give them different types of diet. We give them hepatotoxins, or we genetically knock out specific genes that are important for the normal function of the cells. So in the dietary model, we give them different high-fat diets. We could give them alcohol, or we could give them special diets such as choline-deficient ethionine supplemented diet. We also give them special diet that contains the DDC toxin, or we inject the mice with the CCL4 or CCL4 in combination with the DIN. These are basically genotoxins. Or in the third category, we specifically knock out a gene such as ATG7 or ATG5, which I'm going to discuss in more detail today. And in all of these cases, what we do is we basically harvest the liver tissue, we harvest the blood sample, and we harvest the non-liver tissues other than the liver. So then what we do is basically with the liver tissue, we make a tissue block. Then from the tissue blocks, we use it for a general histology. And we also store some of the tissues at -80. And from this tissue sample, we do different like mRNA or protein expression analysis by standard like cell and molecular biology experiment. Now from the blood sample, we do a serum analysis such as ALT or AST, or analyze specific metabolites that are of interest to our group. And we also isolate these other tissues for inter-organ comparative analysis. So not only this, we do take approach of in vitro as well, where we isolate the primary liver cells by simply perfusing the liver. Here I show the image of the primary hepatocytes that we can easily isolate by perfusing the mouse liver. And we do, after the culturing this primary hepatocytes, we basically do a standard experiment like the one I pointed out here. However, one of the problems with the primary hepatocytes is that after 4 to 5 days of culture, they lose the hepatocyte function and they start to differentiate into a like fibrotic type of cells. So that's why, you know, whatever experiment that we have to do it within that 4 days of time period. And that's why we generally switch on to like another hepatic cell line model that can be continuously grown, and that that they can like that have the future of the hepatocytes. So basically, in today's talk, I'm going to talk about this genetic knockout model where a particular gene such as ATG7 or ATG5s are knocked out in the hepatocytes, and we give a term like autophagy deficient mice. So before I show most of our data about the autophagy deficient mice, let me give you a brief background about the autophagy process. So autophagy process, as Taradai just like give a brief background in a very layman aspect. To me, I envision autophagy process as a dumpster truck that collects all the cellular garbages and make our household stay clean. So, but at a cellular level, what happens is in autophagy, basically it always starts in the cytoplasm by formation of this expanding membrane called phagophore. So this phagophore basically then expands and like forms a double membranous vesicle called autophagosomes, and this autophagosome formation is actually the hallmark for the induction of autophagy. And these autophagosomes can be clearly visualized in the electron microscope as a double membranous vesicle, where we can see the different cytosolic components such as mitochondria or ER or the protein aggregates being enwrapped. And these autophagosome can also be visualized as a punta-like structure in the cytosol of the cell by doing an immunofluorescence staining for the LC3. So after the formation of autophagosome, then it fuses with the lysosome to make autolysosomes, and it is actually inside these autolysosomes where different cytosolic components are broken down into a simpler unit for the recycling purpose. So basically in the field of autophagy, there are two scientists that has been awarded Nobel Prize. One was to Christian Dubé from Rockefeller University in 1974. He was also the one to discover the lysosome and the term, the word autophagy. And the second one was to the Professor Yoshinori Osumi in 2016 for the discovery of different autophagy related genes. However, since 1960 to the 1990s, most of the autophagy related studies were done at a structure and morphological level. However, with the discovery of different autophagy related genes by Professor Yoshinori, then there was a rapid surge in understanding the basic molecular mechanism of autophagy. That means basically people try to like understand different signaling pathways and about the origin of the membrane for the phagophore. So now we basically know pretty much about the different mechanisms that are involved in the induction of autophagy. However, in the field now, people are now trying to understand the pathophysiological role of autophagy in different tissues, and also trying to therapeutically target this autophagy process either by inhibiting or by activating in disease conditions. So my lab is actually interested to understand the role of autophagy in the liver tissue. And in my lab right now, we have 3 different project teams. One is about autophagy in disease, degeneration, and regeneration. I'm going to talk in more detail about this project team today. We also have a research team about role of autophagy in metabolism, and we're hoping to expand our project team on autophagy and liver development after we get our first independent R1 grant. Under the project team B, what we basically try to show is, I mean, in the field, we're trying to push a novel concept about the autophagy and how it contributes to the metabolism. So in the field, what is known is autophagy basically degrades the different nutrients and recycle it, and by that way it contributes to the metabolism. But what we are trying to push a novel concept is that autophagy not only do like that, autophagy can regulate transcriptionally regulate the different signaling molecules such as FXR or nuclear receptors, and directly contribute to regulate the different metabolic enzymes. So that's a kind of, we think it's a novel concept, which I think people may like it, and that's how we are pursuing the autophagy in that autophagy role in metabolism in that direction. So, but I'm not going to talk about this project team yet, but I'll focus on project team yet today, where I try to show what are the pathological consequences that happens in the liver deficient with the autophagy. So that's why we the first question that we wanted to know is what happens when autophagy is defective in the liver. So to answer that question, we first generated this hepatocyte-specific ATG7 knockout. So ATG7 is one of the important genes that is necessary for the autophagy formation. So if there is no ATG7, there is no autophagy process. And I'd like to remind you here that this ATG7 knockout is specific in the hepatocytes, and there are other cells in the liver other than the hepatocyte. So the way we generate this hepatocyte-specific ATG7 knockout is we have an ATG7 flux mice. We basically cross these mice with the albumin-free mice. This albumin-free mice is actually express the Cre recombinase, and the Cre recombinase expression is regulated by the albumin promoter. So we get this hepatocyte-specific ATG7 knockout, and one of the important thing what I would like to pinpoint here is that these hepatocyte-specific knockout mice has intact autophagy in non-hepatocytes or the tissue other than the liver. So the autophagy is defective only in the hepatocytes, which we can see here by a Western blot of the liver lysate, where we don't see the expression of ATG7, and we see the accumulation of a P62 protein, which is actually a autophagy substrate that is accumulated when autophagy is defective. Actually, this P62 is an adaptor protein that is utilized by autophagosome to like phagocytose these different like cytosolic components. So we can see the accumulation of this P62 protein in a just histological analysis here. We saw that there's a good amount of P62 brown staining and the P62 green staining in an immunofluorescent staining of this liver section of autophagy defective liver. So what happens when autophagy is defective is actually many things happen when autophagy is defective. The very first remarkable thing is there is a severe hepatomegaly as shown here by a bigger liver size as compared to the normal one. And the hepatomegaly is because of hypertrophied hepatocytes. The size of hepatocytes are pretty much bigger compared to the normal hepatocytes. Another remarkable like feature of the liver is that there is a severe liver injury. What does that mean is that there is increase in the release of these hepatic enzymes such as transaminases, ALT transaminases in the general circulation. And in this model, the liver injury starts from the 6th week of age. And because of this liver injury, there is severe infiltration of F480 positive like inflammatory cells. We see here more of the brown staining in the liver section of this hepatocyte-specific AT7 knockout. There is increased accumulation of the extracellular collagen fiber. Also increase in expression of the different fibrotic genes such as collagen and TIP1. And these mice spontaneously develop a tumor by the age of 9 months, where we see a lot of tumors being popping out, and the number and size of this tumor gradually increase as the mice gets older. And Because of all of this phenotype, the mice actually has a higher mortality rate. So we have verified this phenotype in ATG5 knockout model, which is another important autophagy related genes. We have verified this phenotype in an indicible model of autophagy knockout. However, one of the important things, what we have noticed during the development of this liver phenotype is that this model reflects all the stages of liver disease, beginning from liver injury to inflammation to fibrosis to the tumor development. So that's why we believe that studying this model will help us, or at least give us a mechanistic clue about how these different pathological changes develop in the context of liver diseases. So that's why the central question for us is actually why liver develop these pathologies in absence of autophages. We believe that it's not because of cells' autonomous failure, because the cells are getting bigger in size, but what we believe is that these are all related to specific signaling factors that are being released by the hepatocytes. And that's our interest. I mean, we really want to identify the factors which is responsible for creating this so many types of liver pathologies. So that's why in relation to that, we have already reported one of the protein called HMGB1, which is actually a nuclear protein. And we have shown that in autophagy deficient liver, the HMGB1 is actually released extracellularly, and they're responsible for the expansion of the progenitor cells, and they're also responsible for the tumorigenesis process. So we have reported that study earlier in 2018. And what we can see here is that in the autophagy deficient liver, there is a massive expansion of different hepatic progenitor cells, as shown here by the staining of CK19, A6, or the EPCAM, which are the specific marker for the progenitor cells. When we genetically co-delete that HMGB1 protein, actually, we don't see the expansion of these hepatic progenitor cells. So that clearly suggests that the HMGB1 is actually playing an important role in the expansion of these hepatic progenitor cells. So it's not only in this autophagy deficient model. Our collaborator in the Columbia University have already shown that the HMGB1 does play a role in other model of the liver injury as well. So in the same study, we saw that this HMGB1 also play a role in the hepatic tumorigenesis. We can see here in the autophagy deficient model, many of the tumor pop out begin from the 9 weeks, but we don't see the tumor popping out at the 9 months in an ATG7 HMGB1 double knockout, suggesting that it slows down the progression of the liver development when we delete the HMGB1. So we have extended this finding in another study where we saw that this extracellular release HMGB1 actually binds to the RAISE receptor that's expressed on the ductal cell, which is also called a progenitor cells. The RAISE also expressed on the macrophages and modulating the tumor microenvironment for the development of the tumor. So we have already reported this study, so that's why we are further extending this study on how we can target this HMGB1 protein to prevent this expansion of progenitor cells and the hepatic tumorigenesis process. But so in this model, so far what we know is actually, we know how this ductal reaction or the progenitor cell expansion happen and how the tumorigenesis happen. We know that these are mediated by the HMGB1 protein. We also know that the liver injury that is happening in this model is because of suppression of FXR and because of accumulation of cholestasis. I didn't show the data about this study today, but what we don't know right now is how this inflammation and how these fibrosis happen in this model. But however, one of the wondering thing that in this model for us is that despite of like severe liver injury that is happening in this model, which we can see here by increasing the transaminases enzyme, which are specifically expressed in the hepatocytes. That means a lot of hepatocytes are dying. But in spite of so many hepatocytes dying, what we see is that still the hepatic functional parameters, such as the serum albumin or the blood urea level, is pretty much normal in these mice. So as we all know that hepatocytes are the ones that are responsible for the synthesis and secretion of the albumin. So that makes us wonder, if so many hepatocytes are dying, how come that the serum level of this albumin is still normal in these mice? So that makes us wonder that something is going on, which we don't know. So that's why we ask this, like one of the important questions, like how could such severely injured liver continue the normal hepatic function? So that's a question that hammer us for a couple of months at the very beginning. And the only thing that pop out in our head is probably in the injured autophagy deficient liver, there might be a new wave of hepatocytes that coming in, and those new hepatocytes is taking on the function of lost hepatocytes. So that's the only possibility that we have it in our mind, and that's why we ask, are there any regenerating new hepatocytes in the injured autophagy deficient hepatocytes? And we took a very general approach. We did a general histological analysis in the older mice of 4 to 9 months old. Since my lab is in a pathology lab, so we have like good infrastructure to do the staining and take a good imaging of those general histological slides. So we took this approach, and one of the surprising things that we noted in this general histological analysis that we see patches of normal looking hepatocytes that are nicely coexisting together with the hypertrophied and inflamed hepatocytes. I'd like to remind you that autophagy deficient hepatocytes always hypertrophied. They are bigger in size compared to the normal hepatocytes. So that really helped us to identify these small size, normal looking hepatocytes nicely coexisting together with the hypertrophic hepatocyte. So this is just a zoom in image just to show you that there are many normal looking hepatocytes living together with these enlarged and hypertrophic hepatocytes. So this is an overlap image showing from region 1 to region 16, just to give you an overview about the extent of the presence of these new hepatocytes in a single section. So after discovery of this new hepatocytes, we basically went on asking, what are these cells? So are these cells new hepatocytes? If they are new hepatocytes, are these cells autophagy competent or incompetent? So that was really interesting question for us, and then that's why we went on to checking whether they are autophagy competent or not competent. And one of the way to check the autophagy competency is by examining the P62 expression level. I mentioned earlier that P62 is an autophagic substrate. So when autophagy is defective, then P62 is accumulated. So that's why we did a P62 immunofluorescence staining in this liver section. And to our surprising, what we found is that there were a lot of areas in the liver section where P62 was negative. Ideally, we were expecting that being an autophagy defective liver, all livers should look green because of accumulation of P62, but we didn't see that case. We see many of the areas of the cells that are negative for P62, and we verify this result by P62 immunostaining as well, where we clearly see the presence of these brown stained P62 positive autophagy deficient hepatocytes nicely living together with these P62 negative normal looking hepatocytes. So we also verify this result in a ATG5 knockout model, which is another autophagy defective model. We clearly see many of the P62 negative areas in this ATG5 liver section as well. This is another immunohistology standing for the P62, again, showing from region 1 to the region 2. There are many of the areas that are P62 negative in this section. So this is a zoom in image just taken from the 4x to 40x magnification. I'm sure you'll pretty much appreciate about the presence of these new regenerating hepatocytes that are autophagy competent. I zoom in the image for this 40x and try to show you with the yellow arrow that there are many normal looking hepatocytes that are clearly coexisting together with this brown stained P62 positive autophagy deficient hepatocytes. So, so far, the conclusion of this study is that there is a new hepatocytes that are autophagy competent appearing in the injured autophagy deficient liver. So after this finding, then we went on taking a very conventional triple WH approach. So basically, we raised 4 separate questions. Question A about when does the new cell appears, and what are these cells, and where are they coming from, and how they are coming there. So these are very 4 important questions for us because it gives us a very important information about these new hepatocytes, about the timing of appearance of these cells, about the identity and function of these cells, and about the location of the cells. Location means where are they coming from? Are they coming from the liver itself, or are they coming from tissue other than the liver? And also it gives information about the factors that are responsible for recruiting these cells into the liver. So we are actively pursuing this this new heparasis in these 4 different directions. So I'm not going to show all the data, but I'll try to show some of our unpublished data about this question A and question B. So I'll start with the question A about when does this new cell appears in this injured liver. So to answer this question, we basically, again, took the in vivo approach. We basically crossed these 87 flocks mice, but this time with the inducible pre-ERT2 mice. So this is an inducible model of ATG7 knockout. So what happens is we generate an ATG7 ERT2 mice. So these mice are pretty much normal because the induction has not been done. So what happens is basically when in a normal situation, these mice express the Cre recombinase, but actually it is trapped in the cytosol because the Cre recombinase is actually fused with the estrogen receptor. So because of the trapping of this like Cre recombinase in the cytosol, the ATG7 is expressed in a very normal way. But however, when we inject the tamoxifen, what happens is this Cre recombinase is actually translocated from the cytosol into the nucleus. And once in the nucleus, then it deletes the ATG7. So until and unless we don't inject the tamoxifen, this mice is a pretty wild type mice. So we have already established this model where we know that we need to inject twice the tamoxifen on day 1 and day 2, and the ATG7 knockout happens on the day 7, and these mice develop the injury by day 15. So basically there is a time lag of 1 week after the ATG7 knockout. So in this mice, we collected the liver tissue at a different time point, and we looked for the P62 negative cells. And to our surprising, what we found is that we saw those new regenerative heparasis only after day 30. So we can see here the presence of a cluster of P62 negative cells on day 30, and the number of cells gradually increase as the mice gets older. And we didn't see the presence of these cells before the day 30. That's why that makes us to conclude that this new heparasis actually arise only on day 30. That means there is a time lag of 2 weeks after the onset of liver injuries. So the injury is necessary for the appearance of this new heparasis. That's the information that we got it from this study of this inducible model. So we also took another approach about crossing this mice with the Rosa mice. This Rosa mice is actually a double fluorescence Cre reporter mice. So we cross these albumin Cre and ATG5 mice with the Rosa mice. So basic idea is that when the Rosa mice is not crossed with the albumin Cre or ATG7, ATG5 mice, all the cell express the membrane-bound tomatodirate at the membrane. Like I saw here in the liver section and the kidney cells, all cells express the tomatodirate in the membrane. However, when they are crossed with the albumin Cre and the ATG5, then they start to express the membrane-bound GFP protein. So the idea is any recombined cell will express the membrane-bound GFP, but any non-recombined cell will express the membrane-bound tomatodirate protein in the membrane. So we generated this heparacyte-specific ATG5 knockout Rosa knock-in mice, and to our surprising, what we found was that there are like membrane-bound GFP expressing hepatocytes, but not only that, there are a cluster of cells that do not express the GFP in the membrane. And actually, these are actually tomatodirate positive, and these are the Merz images trying to show you that how nicely these new hepatocytes that are non-recombined, that are expressing the tomatodirate, nicely coexisting with the autophagy deficient GFP expressing hepatocytes. So this is another image just to show you the cluster of these autophagy competent new hepatocytes coexisting together with these autophagy incompetent hepatocytes. So this suggests that new hepatocytes appear in a autophagy incompetent liver, and this new hepatocyte is paradoxically autophagy competent. So we already characterized these cells by doing a different like cell-specific markers, and so far all of the like hepatocyte-specific markers are expressed by those new hepatocytes. That's why we know that these are indeed actually hepatocytes, not the other cells. So overall, summarizing my talk, so what we know so far is autophagy deficient liver is a perfect genetic model to study the mechanism of liver pathologies, and autophagy deficient liver develops new regenerative hepatocytes that appears after the onset of liver injury. And we know that these new hepatocytes are autophagy competent, And right now, we don't know what is the cellular origin or the factors responsible for the recruitment or the development of these cells in the injured liver. So that's it about my talk. Before I close up, I would like to thank all of my current lab members and also my past trainees. Also special thanks to Tulane School of Medicine for giving a generous startup package, and also special thanks to ASIP and BHERD for giving some research support. So this is my lab team. And before I close up, I would like to make an announcement that there are some lab openings in my lab. If anybody is interested, please do not hesitate to shoot me an email. And finally, I would like to thank you all for your attention. I'm ready to take your question, comments, or suggestions. Thank you.
Thank you so much, Dr. Bilon. It